let's start. So we're going to be learning. I said yesterday we'd be talking about something called microevolution. So, ladies, this is basically evolution at the smallest possible scale. Then we'll talk later on in this unit about what's called macroevolution, which is big change, okay? So this is a little change, and then we talk about big change. So we're going to do a little bit of math today, very basic stuff. It's not really, I don't think it's in your textbook. I think there's like a paragraph on it. So you're not going to find much in your textbook about this, but it's pretty basic math. Yeah, it shouldn't be too hard. So, microevolution. Let's start with a really simple idea. We know that in a population there is a lot of, or not a lot of, but there should be variety, right? And that's important because variety is an ingredient for evolution, natural selection to work on. So here we have a picture, we see lots of variety, right? The problem is if there is no variety, there's nothing to weed out. And evolution needs to weed out things, favor certain things, and weed out other things. Because that's how it works. So we need variety. Variety is very important. But here's something really simple. A really simple idea, okay? An individual cannot have more variety than the entire population. Right? There's got to be more variety in a group of people than in one individual. So we can look at a person here, and we know that for the simple idea of genetics, that we have two copies of a gene, two alleles, one from mom, one from dad. But a person can only have, at most, two different copies, right? Whereas the population could have more than that. So here, for a gene called X, how many types or how many alleles of X do you see? There's three. There's X1, X2, and X3. So population will always have more variety in it than an individual, because an individual can only have two copies of that gene. Okay? The total variety, though, we call it the gene pool. Okay? The total variety is the gene pool of that population. So far, so good? Easy stuff so far? Okay? When we're talking about microevolution, we're talking about changes in this gene pool. Not big changes, but really, really small changes. It could be as small as, right now, if I was to say to you, what's the frequency of X1? How would you do that? How would you figure out the frequency of X1 in this picture? You would add up all the X1s, right? And then you divide it by all of the X's. So I didn't count this. I'm being really lazy. But if I have 10 X1s and I have 100 X's, what would my frequency be? 10. Microevolution is small scale changes, right? So what if we were monitoring X1 and we notice that over time, X1 goes from 10% to 15% to 20% to 30%. It keeps going up. That's an example of small changes because that's only at the level of one gene. Claudia.
Is it like X1 is like 1, or is it like X1 and then X1?
So X1, that's the genotype. Remember the genotype is made up of two copies of a gene? Yeah. So X1 would mean this person is homozygous, and they have both versions of X1. That's what that means.
So you count that as 2 X1.
That would be 2 X1s. Yeah, very good. Yeah. So when we're talking about microevolution, we're literally talking about changes at the level of one gene. Okay? Whereas when we talk about macroevolution, that could be many genes. So we're going to start with a very simple, small picture. Okay? When we talk about changes at the level of a gene, then how do you know if there's change? How would you know? That's what we're going to figure out. Okay? So we know what an allele is, right? Okay, an allele is a version of a gene. And we know what a genotype is, right? A genotype is the code. Okay. So how do you know there is some sort of change going on at a gene. What if we can measure it some way? That's what we're going to do. We're going to measure it using an equation. The equation is called the Hardy-Weinberg Equation. Okay? It's a really simple equation. And the idea is really simple. I'm going to show you mathematically how you can predict the frequency of an allele. Because if you know what the frequency is, then you'll know if it's changing. Because if the frequency is 8%, and every year it's 8%, then that means it's not changing. So if something is not changing, what do you call it? It's in what? What's another word for something that doesn't change? Constant, or another word will be it's in equilibrium. Okay? And that's the opposite of evolution, right? If something is evolving, then it's what? Changing. So if something is in equilibrium, it's not changing. And sometimes actually evolution will favor equilibrium. It will actually discourage change. When would you think it would discourage change? That's usually when the change is what? Yeah, bad. Okay? When the change is bad, it will discourage that. And we'll talk about that later on in this unit. Okay, so we're going to start with some assumptions though, okay? You know how we started genetics and we made an assumption that we have a characteristic and that characteristic is only determined by one gene? And that one gene has two versions, recessive and allele, and then we complicated the hell out of it, right? We're going to do the same thing here. For this, what I'm going to show you, for it to work, you need to make 5 assumptions. For the math here to work, we have to assume 5 things. One is that there is no new mutation. In other words, no new allele. So if we're talking about X1 and X2, we can't talk about a new allele that makes X3 or X4. Okay? Otherwise the math doesn't work. We also have to assume that there's no movement of people. That's migration. We have to assume the population is big. Now, I'll explain later. Not today. I'll explain why we have to assume these things for the math to work. Okay? Because if you don't, if the population is small, this equation actually does not work very well. And we'll talk about that later, okay? We have to assume that the mating is random. That means that if a female is looking at Joe or Jerry, doesn't matter. Otherwise, what's the opposite of randomness? Yeah, it's very good. Selective mating. Means, you look at Joe, oh yeah. You look at Jerry, That's selective mating, okay? When there's no difference between Joe and Jerry, then that will be random mating. And then the 5th thing we're going to assume is that there is no natural selection. Okay? Again, we will come back to these 5 things, and we'll explain how they impact the math. Okay? So, we're only going to start with a very simple picture of genetics again. We're going to go back to genetics now.
Yeah.
All right? Woo! We're going to start with one gene, and we're going to end with one gene. So we're not going to make it actually more complicated than this, okay? One gene, only 2 alleles. The alleles, now, you can use any letter in the alphabet you want, but the letters that were used when this equation was written were the letters P and Q, okay? And P is one allele, and Q is the other allele. So if you have 2 alleles, the frequency has to add up to 100%. So if I have, like, for example, hemoglobin, and there's 2 versions, there's the allele that gives you sickle cell, and then there's a normal one. Those 2 have to add up to 100%. So if the allele for sickle cell is 20%, the normal one has to be 80%. They have to add up to 100%. If there's 3 alleles, then the math doesn't work, because now you have P, you have Q, but they won't add up to 100 because there's a 3rd one. That's why we can only assume 2 alleles here, okay? So these 2 alleles have to add up to 100%. Secondly, if we have 2 alleles, how many genotypes are there? There's homozygous dominant, there's homozygous recessive, and then there's heterozygous. Okay? So this one over here, this is the homozygous dominant. Okay? This is the homozygous recessive, and this is the heterozygous. Now, how is that the case? Well, if I said to you, what is the cross between PQ and PQ? what would you do? Well, you would do Punnett Square, right? And you would say, okay, one individual can donate a P or a Q, and the other one can donate a P or a Q. And if you do that, you get PP, you get PQ once, twice, and you get QQ. Now, in math, PP is the same as P squared. PQ, there's two of them, so there's my 2PQ, and QQ is the same as Q squared. Now, there's only 3 genotypes, and they have to add up to 100%. So, if this is, for instance, if 20% are homozygous dominant, and 20% are homozygous recessive, that means 60% would have to be heterozygous. Okay? So far, so good? Yep. So if this is 20% and this is 20%, then this has to be 60%. Okay? Now, this is the frequency. 1 is the same as 100%. Okay? All right? So, yeah. No. I'm just making up numbers. I'm making up numbers. Okay? Here, the ratio is 1 to 2 to 1, but that assumes that we have an equal number of P and Qs, which is what we assumed in the unit with Mendelian genetics, right? Now we're going to say, because remember, we're talking about individuals. When we did the unit on Mendelian genetics, we talked about Sally marrying Tom, right? Now, we're not talking about individuals anymore. We're talking about a population. We're talking about the gene pool. So we're going to talk about the frequency of the allele, not the individuals anymore. Okay? Because remember, it's not individuals that evolve, it's populations that evolve. Right? Populations. You can't evolve. Alright? Future generations can change, but you can't change. Not in the true Darwinian sense. Okay, so let's talk about flowers. Go back to the example of flowers. Okay? So imagine we have a flower, and the colors are red and white. So red would be, in this case, would be what? Dominant or recessive? Right, so this is dominant. So we have dominant. So it could be homozygous dominant, or it could be heterozygous. Whereas if you're a white flower, what are you?
White.
Well, what are you? Thanks, I know. So what are you, though? You're what? Homozygous recessive, Q squared. So you can spot a problem here. Because if you're a red flower, you don't really know if that flower is, or that plant is homozygous red or heterozygous. Because in both cases they look what? They look the same. But if you have a white flower, because white is recessive, then you automatically know that what? That plant must be what? Homozygous recessive, right? So here, this is just a simple example where you have, like, what's the allele frequency for red and white? These are just made up numbers, so don't take them seriously, okay? So if I have 320 flowers that are homozygous dominant, now here, if, like, you wouldn't know they're homozygous dominant. These two flowers would look exactly the same, okay? So a question in real life, you would not know. Here, it's broken down to you, so it's unrealistic. You're actually being told that we have 480 flowers that are red, because 320 and 160 add up to 480. And we have 20 flowers that are white. So how many flowers in total? 500, right? Or 500 plants. Now, we have 480 that are red. Here we're told that 320 are homozygous dominant and 160 are heterozygous and 20 are white. And then they say, well, if you want to know what's the frequency of red and white, you can do some really simple math. You can say, okay, look, if you're homozygous dominant, how many red alleles do you have as an individual? Homozygous? If you're homozygous, you have 2 red alleles, right? So if you're an individual with 2 red alleles, and there's 320 of you guys, then how many red alleles would they have in total? 640, right? 320 homozygous red individuals would have 640 red alleles. Whereas 20 homozygous white individuals would have 40, because each individual has how many white alleles? 2. Remember, they have 2 little r's, right? So how many little r's would 20 people have? Sorry, not 20 people, 20 plants. 40, because 20 times 2. Now, if you're heterozygous, how many copies do you have? You have 1. So 1 heterozygote would have 1 red allele and 1 white allele. So how many would 160 heterozygous red flowers have in terms of red alleles? 160. And they would also have 160 white alleles. The math is really simple. If I have 500 individuals and each individual has 2 alleles, how many alleles do we have in total?
Say that again.
If I have 500 individuals and each individual has 2 alleles, you would have 1,000. So that's why it's divided by 1,000. In this case, the frequency of red is 80%, because I have 800 red alleles. And the frequency of white is 20%, because I have 20 out of 1,000. Sorry, 200 out of 1,000. Sorry, 200 out of 1,000. It's 20%. The math is easy, but you don't need to do this. I'm going to show you a different way you can do this, okay? So I'm going to show you an equation. So do you recognize this? What is this? It's a Punnett square, just it's on its side, right? The diamond. The diamond is a square, basically tilted. So I'm going to show you how you can calculate using the actual Hardy-Weinberg equation, okay? So I'm going to go back to the example of the flower. How many individuals do we have? We had 500, and 80% were red, and 20% were white alleles, right? So let me show you the math behind that. So what do we have in that situation? 20 out of 500 flowers were what? White? Okay. And 480 were red, right? Okay. So I'm going to show you how you don't have to do, because remember, in real life, you can't tell by looking at a flower what its genotype is unless it's recessive. If it's red, you can't tell if it's homozygous or heterozygous. But there is a way to figure it out. It's really, really simple. And this is what we're going to do. We're going to use this equation. Okay, that's one. And we're going to use this one. Okay, now remember, this is the frequency of what? This is the frequency of the alleles. Okay, and this is the frequency of the genotypes. How many alleles do we have in that scenario of the flowers? There's 2, right? There's a red allele and there's a white allele, okay? Let's change the color. Okay, so which one's dominant? Okay, so I'm going to make, because usually we say the P is the dominant allele, so we're going to say the P is the red one, and the Q is the white one. Okay? So far, so good. And we know that the red and white have to add up to 100%. And we already know what the answer is. We know it's 80%, 20%. I'm just going to show you how you can do it using an equation, because you won't really know what the composition of the population is going to be. So, here's what we know. We know 20 are white. 20 are white. And we know for you to be white, you need to be homozygous what? Okay. For you to be homozygous recessive, that is your genotype. Correct? Okay. So, here, genotypes. There's p squared, there's 2pq, and there's q squared. Which of those three represents the white condition? Q squared. So this is what we know. We know then that 20 out of 500, right, has to be my q squared. The frequency of my white individuals is q squared because q squared is my recessive genotype. White is recessive, so q squared is my white one. So now we just do some simple math. Can I get rid of the square? Yeah, I can do the square root. Okay, so what's the square root of 20 over 500? So here's the thing. If my white, so remember, the Q, the P and Q are the alleles. The P squared, the 2pq, the Q squared are my genotypes. If I take the square root of Q squared, I get my Q allele. What's this saying? It's saying that 20% of my alleles are Q, which is white. The other must be what? 80%. And that's what we got, right? That's what we got with our flowers. 80% were red, 20% were white. But the problem is, the way it's laid out here, you're not going to get this in real life. You're not going to get the breakdown of your homozygous and heterozygous. You might, but you might not. So the way you figure it out is by using this equation, the Hardy-Weinberg equation. So let me show you another example using the exact same logic. Remember our taster lab? Okay. So imagine we have 200 people. Out of 240 people can taste PTC. Or we can say sodium benzoate, because it's the same genetics. And we want to know what the frequency of the taster allele is, okay? So there's two alleles, right? If you remember, there's the allele for tasting. We'll call it the taster allele. And then there's the non-taster. Who was recessive? Yeah. This is dominant. And this one was, okay. So for you to be non-taster, what would your genotype have to be? Yeah, it would have to be Q squared, right? For you to be a taster, you could be homozygous dominant, or you could be heterozygous. But for you to be a non-taster, you need to have two recessive alleles. Okay, so this is what we know. We know that 200 out of 240 can taste. Now, Claudia, yeah, you would. You wouldn't use this information. This information is actually, yes, what's relevant is how many who can't taste. Now, why, Claudia? Exactly. Look, we have 200 people that can taste. For all you know, that could be 200 homozygous dominant individuals. It could be 200 heterozygous. We don't know. But this is what we do know. We know that 40 can't taste, and we know that they must be Q squared. So that's what we start with. You always start, listen carefully, always use the recessive, okay, or the Q squared. Never get tricked and use the dominant allele. Always start with the recessive one. So let's solve for Q squared. Q squared is 40 over 240. And again, we take the square root. So what's Q? 0.5. 4, we'll just say 4-1 then. Okay, round off to 4. So here's the thing. My Q, which is non-tasting, is 41%. P plus Q has to equal 1. So P must be 1 minus Q. Well, because they have to add to 1. So if Q was 0.41, P must be 0.59. These two numbers have to add up to 1. Right? To 1. Miriam. You can predict it now. I'm glad you brought that up. You can actually make a prediction. Now, this question does not ask you for that. All it asks you for is, what is the frequency of the taster allele? But if I said to you, okay, what's the predicted frequency of your homozygous dominant? Imagine I asked you that question. Said, well, homozygous dominant would be P squared. We just figured out what P is. We know it's 0.59, right? No, it wouldn't be the same. We just figured out that P is 0.5. Q is 0.41. P is 0.59. So if I want to know how many were actually homozygous dominant, it's really simple math. I know that 1P is 0.59, but the homozygous dominant, which is P squared, would be 0.59 squared. So what would that be? What's 0.59 squared? Yes. What is it, Kiara? So 0.3, we'll just say 0.35. So 35% of that population would be homozygous dominant. And like Miriam just said, if I want to know what my heterozygous frequency is, well, that would just simply be 2 times P, which is 0.59, times Q, which is 0.41, and that would be, what is that? 6, 22, about 48%. What is it? Should be close to 0.48. Is it? So 0.48. Just well, because this is close to 6, this is close to 4. 6 times 4 is 24. 24 times 2 is 48. So it now has to be 48. There you go. Yeah. All right, let's do one more. Yes. Yep. Okay, so what's Q squared? But Q squared was 40 out of 240, right? So 40 out of 240 is 1/6, which is 0.16. Now, if I know if I add these three up, it should be 1. Because P squared plus 2PQ plus Q squared equals 1. But they might not add up to 1. They might add up to 0.99 or 1.01 or 1.02 because of what? Because of the way you rounded it off. Okay? But it should be very close to 1. So let's do one more example, okay? Remember the mods? Remember there are two types of mods? The dark and light one? Yeah, it's almost too hot. Remember there's two types of mods, right? There's a dark one, and then there's the light one. The dark is dominant. So the dark will be P. And the light is Q. Now in this case it's 500 and 500. So 500 are light. What would the light be? What would the genotype for the light be? So Q squared would be the frequency of the light allele, which is 500 individuals, and there are 1,000 in total, right? So if I want to solve for Q, it would be the square root of 500 over 1,000, which ends up being what? It's the square root of 0.5. 0.71. So 71% of my alleles are light. That means my dark allele has to be 0.29 because again, this plus this have to add up to what? To 1. Okay. So how is that? Is that like really big, really difficult? Okay. Well, this is what we'll talk about next time. How do you know that there's change unless you know how to monitor the change? For example, if I said to you, here's a drug that will change your heart rate. How do you know it changes your heart rate? What do you need to know? You need to know what your breathing heart rate is, right? You need to have some way to measure the change in the heart rate. So this math will allow us to measure allele frequency so we can monitor whether or not that allele frequency is changing in a population so we know that it's microevolving, for instance. Okay? So we need to be able to monitor the change in some way. All right, ladies, that's where we'll stop off for part 1.